The presentation is focused on a phase 1 clinical trial for CAR T-cell therapy for solid tumors, more specifically for mesothelioma, pleural mesothelioma, metastatic lung and breast cancer to the pleural cavity. So we know CAR T-cells had showed great promise and results in leukemia and lymphoma, blood cancers, but the solid tumors to date, the results are not good at all. So based on that, we conducted a phase 1 study. This is targeting a mesothelin, a protein expressed on the cancer cells with very low levels on the normal tissue. And that's what we did in this study. So we first investigated the mesothelin expression levels in the cancer cells and the normal tissue. And obviously it's overexpressed on the cancer cells and imparts aggressiveness to the cancer cell. So we decided that's a good antigen to target. We created a second generation mesothelin-targeted CAR, chimeric antigen receptor. And we did studies in the mouse because of the pleural cancers are about 150,000 patients per year in the United States. So we gave systemically through intravenous, that's how CAR T cells are given for leukemia and lymphoma patients. But instead we also gave directly into the chest cavity, pleural cavity. What we showed is even when we reduced the dose by 30-fold, when we gave directly into the chest cavity, the CAR T cells are activated, proliferated, and reached a higher level, and the efficacy is much better. Based on that, in this phase 1 clinical trial, it's a dose escalation trial with cohorts with cyclophosphamide preconditioning. We gave directly into the pleural cavity in patients with mesothelioma, lung or breast cancer with pleural metastasis, once we confirm that the tumor is overexpressing mesothelin. And we followed these patients once a week for 8 weeks. There is some concern about on-target off-tumor toxicity because mesothelin is expressed at very low levels in the pleura, peritoneum, and pericardium, but we did not notice any on-target off-tumor toxicity to date. While the phase 1 study is being initiated, we mimicked that in our mouse model. In our orthotopic pleural cancer model, we established a large tumor burden and purposefully gave a very low dose of CAR T cells. What we observed is CAR T cells are reside in the tumor, but they are functionally exhausted. In the presence of overwhelming tumor burden, CAR T cells overexpress PD-1 and the tumor cells express PD-L1 and L2. And we are able to rescue the exhausted CAR T cells and prolong the functional persistence if we give the checkpoint blockade anti-PD-1 agents. So based on that, in this clinical trial, in the later cohorts, after patient received a single dose of CAR T cells intrapleurally, once we ensure that there is no toxicity for about 6 to 8 weeks, we started these patients on anti-PD-1 agents, and a combination therapy of 11 patients. In 8 patients, we saw responses, a response rate of 72%. 2 patients complete response, 60 weeks and 38 weeks, with no additional treatments, and about 6 patients partial response, some of them for a year, not requiring any other treatment. In pleural mesothelioma, these results are encouraging because the last FDA-approved therapy for mesothelioma patients was in 2003, a platinum combination, cisplatin pemetrexed, that showed a survival benefit of 3 months in a phase 3 randomized trial. So obviously this is a phase 1 study to determine the safety, but we are encouraged by the early signs of efficacy with the combination therapy. So to date, we are starting the anti-PD-1 checkpoint blockade from week 6 or week 8, but now we are applying to IRB and FDA to be able to start that early by week 3 or week 4. Thereby we can enhance the efficacy. On the other hand, anti-PD-1 agents has to be given every 3 weeks, perhaps for the rest of the life for the patients. And also anti-PD-1 agents may have a non-specific immune adverse events in non-tumoral areas. With that in mind, we genetically engineered a PD-1 decoy receptor, PD-1 dominant negative receptor, where the extracellular receptor is kept that binds to PD-L1 and L2, but we took out the intracellular signaling, thereby the CAR T cell is no longer inhibited. So we now put this PD-1 DNR in CAR T cells, and we published in JCI preclinically that the results are encouraging, as good as giving anti-PD-1 agent. But more importantly, the PD-1 DNR is within the CAR T cell, so it's tumor targeted as well, avoiding any non-specific adverse events. So we are now conducting IND studies to be able to take it to clinical trial early next year. Again, it's a phase 1 study, so we are happy to prove the safety and early signs of efficacy. Obviously, we need to follow long term to establish the anti-tumor efficacy.